Welcome back, everyone, and welcome the following panel, where the wild things are conserving Colorado's endangered and sensitive species. And yeah, please welcome.
Hello. Yeah, thanks for coming to our presentation this morning. Brandon, Corrine, Julie, and I are all master's in ecology students here at the school, and we're researching where the wild things are in Colorado. The common thread tying our projects together are conserving Colorado's endangered and sensitive species, where we evaluated occupancy, habitat selection, and resource use to help conserve our study species in the face of continued threats of habitat loss, fragmentation, wildfire, and climate change.
Colorado contains many unique habitats that are drastically impacted by the aforementioned factors. Mature conifer forests are under increasing threat from more frequent and severe wildfires. Climatic changes in high elevation habitats are providing greater opportunity for diseases to spread. High elevation spruce fir forests are being decimated by spruce beetle outbreaks and threatened by wildfire. And alpine environments are experiencing warmer temperatures and earlier snowmelts.
Next slide, please. So this kind of just outlines the project areas where we worked. And next slide. I'm Brandon Skerbetz, and for my project, I investigated Mexican spotted owls in south central Colorado. These owls rely on narrow canyons that contain mature mixed conifer forests that were historically logged, and this led to the owls getting a federally threatened status. But now intense wildfires pose a major threat to these habitats, and public land managers only have a vague idea as to where these owls occur across Colorado. And so for these reasons, I expanded consistent search efforts to encompass all of the Mexican spotted owl critical habitat unit, which is shown on the left there, to identify what characteristics of the habitat are influencing their occurrence. And I then created a predictive map using these habitat features to identify potential locations of the owls that we may not currently know of. Next slide.
Hey, I'm Corrine. Can everybody hear me okay? Awesome. Okay, so my name is Corrine Ryan. I am also an MS in ecology student. This is my last semester here at Western. And my thesis project was on determining the foraging habitat requirements of sensitive bat species in high elevation habitat. So this was done in the Gunnison Basin and surrounding area. You can see my survey sites in that map on the left. And then on the right, that's an example of one of my really beautiful survey sites. So bats are globally distributed. They make up 20% of all mammal species, and they perform billions of dollars a year in ecosystem services just in the United States. In addition, they're under many threats, including disease, climate change, and habitat loss. And they're also severely under-researched. So they require more research in order to create informed management and conservation plans for them. The disease that is kind of central to my research question is something called white nose syndrome, which is currently in the process of just decimating bat species all over North America. And so I saw this need for further research to inform these conservation plans for these species that are susceptible to white nose syndrome. So I'm looking at 4 different habitat covariates that may influence habitat selection for these bat species to see if we can kind of figure out better how they occupy these high elevation habitats in Western Colorado. Next slide.
Hi, so I'm Melissa for my master's thesis research. I evaluated habitat suitability for Canada lynx and habitat selection by snowshoe hares, their main prey, in the North Powderhorn Wilderness here in southwestern Colorado. The Powderhorn spruce fir forests have seen extensive mortality due to spruce bark beetle outbreaks over the past 2 decades. And so as a result of these outbreaks, these forests have lost the majority of their mature trees in the landscape. The primary goal of my study was to evaluate severe beetle impacted areas for lynx habitat and hare habitat selection to help inform management decisions in the area to simultaneously reduce a dangerously high fuel load, which contributes risks of severe wildfires, which can threaten nearby communities, as well as any remaining critical wildlife habitat in the Powderhorn. while also encouraging the regeneration of healthy spruce fir forests, healthy habitat for these two wildlife species. Next slide.
And my name is Julie. For my thesis, I assessed the floral nectar sugar available to the Uncompahgre fritillary butterfly based on the butterfly's flower selection in order to gain an understanding of the food resources required by this butterfly as an adult and to characterize one of the aspects of the butterfly's preferred habitat, to have a better understanding of why the butterfly occupies certain areas and certain ranges and why it doesn't occupy others. My thesis was near the Lake City area in the San Juan mountain range, so not too far from here. Next slide, please.
So as you've probably kind of figured out by now, the overarching goal of all of our projects is to inform management actions to best conserve Colorado's wildlife and its habitat. And so without further ado, let us introduce you to Colorado's wild things, starting with Brandon.
Next slide. This one's actually in this slide. Oh, yeah, that'd be great. Thank you.
Yeah.
All right, so with the Mexican spotted owls, my project was in coordination with the Bureau of Land Management Royal Gorge Field Office based in Canyon City, Colorado. And one of the major components of my thesis research that I'm presenting on today involves the modeling and predicting of Mexican spotted owl occurrence in South Central Colorado. But I wanted to start the presentation by thanking my committee members for their huge contributions, both towards the project and for my professional development. Patrick McGee, Madeline Vanderkirk here at Western, and then Matthew Rustan and Jordan McMahon with the BLM, as well as the funders for this project, the Bureau of Land Management, RGFO, Colorado chapter of the Wildlife Society, and Western Colorado University. And last, I want to pay tribute to and honor the Mexican spotted owl for providing such unique and impactful memories for me over the last 3 years. So long live the Mexican spotted owl. And so to start off, I wanted to provide some brief background. I skipped again. So the Mexican spotted owl is one of 3 subspecies of the spotted owl, and they're distributed in the southern regions of the United States as well into Mexico. And you can see as they get into the southwestern United States, they become pretty patchily distributed. And they have, I mentioned this before, but they're federally threatened, and this status was gained in 1993. Their Mexican spotted owls rely on very specific habitat variables. So they like really narrow canyons that contain mature mixed conifer forests in the bottoms. And the combination of that habitat becomes pretty rare to find. And that's what describes their patchy distribution as they move northward into the United States. And the logging of these old growth or mature growth forests is what initially caused their threatened status. But now, high severity stand replacing fires are the largest threats to their habitat. And that's in the face of climate change with increased droughts. And if we focus in on the spotted owl or Mexican spotted owl habitat in the United States, you can see that the USGS has predicted very high wildfire hazard potential. So this just kind of shows us that it adds evidence to a study that found since 1986, Mexican spotted owls have lost around 21% of their habitat, and 8 to 35% of that is due to wildfire. And so to try to understand their distribution better in Colorado, I expanded search efforts beyond 6 historical survey sites where we know the owls are, and tried to survey the full critical habitat unit that's between Canyon City and Colorado Springs to try to answer the question of where else are resident Mexican spotted owls in this critical habitat unit. I also wanted to identify the habitat features that are influencing the owls occurring there and use those to predict where the owls might be across this full habitat unit. And the way that I was going to achieve these objectives was by using passive acoustic monitoring. I want to touch briefly on what occupancy analysis and modeling is. So whenever you go outside and you're looking for an animal, there's a lot of times that you might just walk by the animal and you don't realize it's at the site that you're surveying. So you'll say that that site is unoccupied. But once you repeatedly survey these sites, you're able to account for or predict the detection probability. And this allows you to then predict the true occurrence of the species across a larger area. And you can take this further with occupancy modeling by figuring out what type of variables are affecting the detection probability, as well as what's affecting the species occurring in a spot. And so that's what we did for this study. And so from March 14th to August 11th in 2022, I expanded consistent search efforts to this full critical habitat unit, which was about 278 square miles. And the way that I selected survey sites was by tessellating 300 hectare hexagons, which are close to the home range size of these owls across the full critical habitat unit. I got rid of survey sites that had a mean elevation less than 90 or above 9,500 feet, because the owls typically don't occur above 9,500 feet. And then I got rid of cells that had less than 70% of public land ownership. That way I could actually fully access the survey site to look for the animals. And so then I was left with a list of surveyable cells, and I used a balance acceptance sampling order to choose the order to survey these cells and which sites to visit. And this sampling design ensures that all of the sites are spatially balanced and randomized so that I can make inferences about the entire critical habitat unit. And I knew that there were going to be a lot of survey sites that I probably wasn't going to be able to access just because the topography of this place, it's a really complex canyon network, and accessing some of these sites was just going to be impossible without like a helicopter or some rappel equipment. So I was able to sample 43 survey sites using 3 ARUs, or autonomous recording units. They're just recorders that you schedule to record all night. I scheduled them to record 1 hour before sunset all the way out to 1 hour after sunrise. So it was all the way through the night. And they were left at each survey site for 7 nights of recording. And within the survey cells, I made sure to keep the detectors at least 250 meters away from the boundaries of the cell so that I wasn't getting detections of owls from outside of the survey cell. And then also the detectors, I made sure to keep them at least 500 meters apart from each other so that they weren't overlapping each other. And I was effectively surveying as much of the survey cell as I could. This map kind of shows the status of the sites that I was able to visit. The light blue sites, there's 29 of them that I ended up having to skip. So this kind of just shows that it was a good thing that I used that balance acceptance sampling design, because I did have to skip a lot of sites due to topography like this. Sometimes you're walking up a canyon and there's just a waterfall and there's no way to get around it. And so the sample design allowed me to work around that and still have the spatial balance for my inferences. And so once I collected all of my acoustic data, it was about 2 terabytes worth of data. I ran it through an acoustic analysis software that I trained to look for all Mexican spotted owl vocalizations. The most common Mexican spotted owl vocalization is the 4-note call below. And this was kind of the main one that we were looking for. From the 43 survey sites that I surveyed, 9 of them were occupied by the owls. And I ended up capturing 672 male vocalizations and only 216 female, and no juvenile vocalizations. One interesting thing is that the red sites are the occupied sites. And whenever we compare those with the historical sites, which just popped up in the white, all of the occupied cells are proximal or adjacent to those historical sites. And so this kind of gives evidence that we mostly know where the owls are across this whole area. And they're all in the southern region on BLM lands. But moving on to the occupancy analysis, it estimated that only about 21% of this critical habitat unit is actually occupied by the owls. And whenever you're in one of these survey cells and an owl is there, there's only about a 51% probability that you'll actually detect them. looking into what features are influencing the owls occurring on the landscape, I did a model selection process. And for the simplicity of it, what we found as the factors that are most influencing the owls on the landscape are mean slope. So that's kind of explaining the topography. And then the evergreen open tree canopy and mixed evergreen deciduous open tree canopy. So mixed conifer forest with open tree canopy. And whenever we break these effects down individually, we see that mean slope has a really strong positive effect. And so this is kind of just supporting that canyon network where the slope is really high is helping to contribute to these microclimates that the owls are selecting for. And then the next covariate was the evergreen open tree canopy. There's a lot of literature that supports this also, but it says that as the percent cover of the evergreen open tree canopy increases, the predicted Mexican spotted owl occupancy increases, and that's likely because they're using these more open spaces to fly and forage in, rather than the dense forest that they prefer to nest in. And lastly, the last covariate was the mixed conifer forest with open tree canopy. That had a negative effect. And we think that that's because in our study area, this habitat type is associated with more riparian areas that are open, and that's contributing to a warmer microclimate that the owls are avoiding, as well as this area has a much less dense understory, and that's not supporting for their primary prey source, which are wood rats. And so once I was able to identify those three features in the top model that are best predicted to be influencing the owl's occurrence, the evergreen open tree canopy and the mixed deciduous open tree canopy, that's this map here, and then the mean slope. I extracted the values for each of these for every survey cell across the whole critical habitat unit and used the combination of those to predict the probability of occupancy for the owls at that site. And so what we found was that there's a core area in the southern region, pretty much where the owls are occurring, that have high predicted occupancy probability. But there are a few fringe locations to the west and then one to the east that would be really interesting to go look into and see if there's owls that we didn't previously know of in these locations. And so looking forward, the BLM in Canyon City is looking to use these passive acoustic monitoring techniques in tandem with their active nocturnal night surveys that they do, in effort to try to locate and detect new owls, including juveniles. If there's any land actions proposed in the critical habitat unit, they can use this work to try to identify if owls are present. And then also consecutive years of occupancy modeling will allow the BLM to understand the colonization and extinction of sites. And so they'll understand if the population is growing or stable or lowering from that. The predictive map will be helpful for trying to find if there are new owls in the location, and then also to try to target areas to reduce fuel loading in habitats adjacent to their core areas. And lastly, these methods are useful for any species that are vocally active and orally identifiable. And there's a lot of other analyses that you can do beyond this occupancy. So with that, I don't know if there's any time, but open the floor for questions, and thank you all.
What does your office say?
Yes. Sorry, there's. Go ahead, Jules.
So first of all, that was amazing, and this is great work you're doing. And it seems super thorough and awesome. Did you learn how to use the software you used to make those models here at Western, or was that from previous experience?
It was from here, as well as just kind of a lot of. I've worked with like a few other species that I've done occupancy modeling for, and so yeah, it was kind of a personal like adaptation of what I've learned in class and then just specifying it for this specific analysis. So yeah.
Brendan, please remember to repeat the question.
Oh, I'm sorry.
That's for everyone.
Okay, thank you.
Here's a question on Zoom. Andrea Williams wants to know if there's any theory for why there were no juvenile owl vocals detected.
Any theory for why no juvenile owls were detected? Yes, I think that's because I expanded my search efforts to like try to cover a large area. And so I don't think that I ended up placing my detectors in any like core areas or near nests where I would be able to detect juveniles. I feel like the majority of my detections came from more foraging habitat, and we kind of see that when comparing it to the historical locations.
Sydney, one.
Yeah.
Similar to that question, do you have a theory why there were so many more male owls than female?
Yeah, why is there more?
Repeat your question, please.
Yeah, why is there more male vocalizations than female? That is typical, like all of the vocalization studies find that the males do vocalize more than the females, but there's also just a wide variety of vocalizations that both sexes use. And I think that this also kind of ties into finding owls in the foraging habitat. It's probably more likely the owls that are dispersing out and trying to find food for potentially the females that might be on a nest.
Well done.
Thank you. Appreciate it.
Okay, I'm going to try to share my screen here. Please let me know if it works or not.
If we need to pivot on the screen.
Okay, are you guys seeing the first slide of my presentation, or are you seeing the presenter view?
Presenter view.
Okay, perfect. Let me, how about now?
Perfect.
Awesome. Okay, so my name is Corrine Ryan. I am an MS in Ecology candidate at Western Colorado University, advising under Madeline Vandekirk, and my thesis project has been determining the foraging habitat requirements of sensitive bat species at high elevations in Colorado. So I wanted to start with a quick land acknowledgement, just to acknowledge and respect the Ute tribe, which is part of Western Colorado. It encompasses their traditional and rightful lands. I'm not going to read out the whole land acknowledgement. You guys can go ahead and read that on your own. But I wanted to just take a moment to respect the tribe that was here before us. Okay, now moving on to my individual acknowledgements. These include all of the professionals that have provided guidance, advice, help in the field, lending technical equipment and expertise. I could not have done this project without any of them, so sincere thank you to all of the people on this slide. So for some background, obviously bats are the coolest flying mammals. They're also the only flying mammals, but we don't need to talk about that. They are globally distributed on every continent except for Antarctica. They make up 20% of all mammal species, so 1 in every 5 mammal species is a bat. They perform key ecosystem services such as pest control, pollination, and seed dispersal, and they provide billions of dollars of economic services in the United States alone every year. They are also facing increasing threats such as climate change, habitat loss, and disease. And they're severely understudied. So pretty much every bat species, especially here in North America, requires more research in order to get to know them a little better. So this map is the current spread of white nose syndrome as of early 2022, which is when I started this project. White nose syndrome is a disease that has been absolutely decimating bat populations of several different species from when it was introduced to the United States back in 2006. You can kind of see based on the color coding that it started over on the East Coast, and since then it's been progressively spreading west and kind of creeping towards Colorado. And so at the time that I started this project, we knew that it was an imminent threat in Colorado, and so we wanted to gain as much information as possible before it entered the state. Unfortunately, as of July of last year, white nose was officially found in Colorado, and it has continued to spread across the western states. So you can kind of see the difference between these maps, and this is literally within 6 months of each other. So this is a disease that spreads super quickly. There is no current cure or treatment. So it's really, really important that we try to study and conserve these bat species that are impacted by white nose syndrome. So this is kind of where my research question comes in. There is a clear need for further research to inform conservation plans for these particular species that are susceptible to this disease, white nose syndrome. And so I wanted to look at the habitat level variables that influence whether or not certain bat species are present in particular areas. And so my research question was, how do water body type, land cover, elevation, and human footprint impact the occupancy of sensitive high elevation bat species in Western Colorado? So on the bottom left here is a map of my study area. Those squares that you see right there are part of a protocol developed by the North American Bat Monitoring Program, which was created in about 2015. It's essentially this continent-wide consistent monitoring protocol in which they divided the entire continent up into grid cells. And so that's kind of what you see that are the blue squares on the map. So I selected 8 of those grid cells for survey. And then what you do after that is you select 3 to 4 survey points within each grid cell to survey through passive acoustic monitoring, just like Brandon did. And there's a good example of what a monitoring unit looks like on the bottom right over there. Basically, the way that it works is you set those up in an area, put them above any clutter, which is, vegetation or anything that the sound might bounce off of. And every night they would come on at about sunset, go off at about sunrise. They sit there and they record every time they hear a bat fly by and call. So every time a bat yells at the microphone, it picks it up, it records that. And so these are some of the survey sites that I was lucky enough to be able to set up camp in. You will notice that they all have water sources because that is one thing that's pretty consistent for the NABAT protocol. You have to have a water source nearby to set up these recording units because it's just kind of a given at this point that for most bat species, especially insectivorous bats, which is what we have in Colorado, water sources are pretty imperative to their survival and foraging. So I had a variety of water sources that I set them up near: streams, rivers, lakes, reservoirs, things such as that. As well as, a variety of elevational gradients as well. And you can see my field technician in 2 of these pictures over here. She was also very crucial to the success of the project. So once I had all of this acoustic data collected, I believe it was just over 55,000 calls that were recorded. So quite a lot of data. I plugged that into a program called Sonobat, which, as you can probably guess from the name, is specific for acoustic analysis of bat calls. And what it does is it'll take those calls like you see on the left over there, and it'll run it through its acoustic identification software and spit out the species name or what it thinks the species that made the call is. So you can kind of see the difference between these 2 calls, between the top and the bottom. The first one is a lot longer, way more straight up and down. The bottom one kind of has more of a foot. We call that a dog leg type shape. And so those are from 2 distinct species. So once the program is done automatically classifying all of these calls, I go through and I manually verify a subset of those calls just to make sure that it's doing its job the right way. It didn't get confused. There weren't, multiple species in one call, sound file, that kind of thing. So as you can imagine, it's a long process, big learning curve, but it's actually pretty fun once you get into it. And then for my actual covariates, my variables, the human footprint, water body type, land cover, and elevation that I mentioned before, I conducted spatial data processing in QGIS. So I extracted the spatial data from there in order to use the data for those 4 variables for my analysis, which was done in R. I also did occupancy modeling, just like Brandon. He provided a really good explanation of that, so I won't really go into it. But essentially you can plug in, the presence and absence data for these species, as well as any variable data that you think is relevant. And the occupancy modeling will spit out what it thinks is the top performing model. And so it can tell you which species are associated with which covariates. And so these are the 14 species that I was able to find within my study area, which is pretty incredible considering that there are 19 total bat species in Colorado. So I was able to find 14 out of 19 of those in this high elevation area. And I just wanted to throw up some pictures of them because I think they're all absolutely wonderful. Go ahead and pick your favorite. And so these are my 10 study species. So these are the ones that have been shown to be susceptible to white nose syndrome. And so they're at a particular risk. And so these are the species that I wanted to focus on. So moving on to my results, I found some really interesting stuff. So when I was looking at elevation, I expected to find that certain bat species would be present, at particular elevations. I expected to find a higher biodiversity of bat species at the lower elevations as opposed to higher. But I really didn't find that. And I was kind of curious about why. So I found 4 species above 10,000 feet, which was higher than they have ever previously been recorded. 3 additional species were found much higher than previously thought. And 4 of these study species were also found outside of their current estimated ranges, which is also because of that high elevation habitat. It's thought that there really wasn't a whole lot of suitable habitat for these species in these areas because of the elevation. So these are the 8 study species out of my 10 that actually had enough data for me to be able to conduct analyses. I conducted my study during 2 separate survey periods, kind of one in midsummer and then one in the early fall period. So I included an analysis for seasonality that you'll see up there at the top right, just so that I could potentially account for any variation. Maybe there's some kind of migration pattern going on that I would be able to pick up through that. As you can see in the table, the only significant result that I had is TLCI, which is that human footprint covariate. And that was with the small-footed bat, which is the little guy right here outlined in red. So what I found is that the small-footed bat had a significant relationship with human footprint. As you can kind of see in that box plot on the left, there is occupancy on the x-axis, 0 meaning unoccupied, 1 meaning occupied. And it's pretty clear just from looking at this that the occupied sites had a much lower human footprint than the unoccupied sites did. And so this would indicate that this particular species really tends to avoid areas that have a high human footprint on the landscape. So, human alterations such as buildings, roads, resource development, that kind of thing. So we can draw some conclusions from this. First of all, many bat species are present at higher elevations than previously known. And that's really interesting. And it seems like there could be 2 basic explanations for that. It's either maybe they tolerate high elevation climates better than we thought because those areas do tend to be understudied. So maybe they were present there all along and we just didn't know. Or the second explanation is that high elevations are warming. And so their thermoregulatory capabilities are less stressed in these high elevation areas now that they're becoming warmer. And so bats are moving up in elevation. So there definitely needs to be further study and investigation into that. The second major conclusion is that, like I said earlier, small-footed bats are highly impacted by human footprint, as illustrated by this really lovely, well-done graphic on the right. In addition, it seems like I'm probably missing a pretty important habitat variable given my lack of significant results. So there could be something else that we're missing here that could account for the variation in occupancy in this particular study area. And so that kind of leads me to thinking about questions for future research. It would be really interesting to look at comparing occupancy records with temperature trends within these study areas to see, if temperatures are in fact increasing in accordance with bat occupancy here. I would also want to look at the factors of that human footprint metric that most impact small-footed bat occupancy. So those can include, like I said, things like land use type, roads, structures, resource development, that kind of thing. We would also want to look at how white nose impacts bat species at these higher elevations, because again, that's another area where we really don't have a lot of research, and it is a landscape where they're particularly sensitive because they're so limited. Like there's really only so high they can go up, right? And so we want to look at how disease impacts these particularly sensitive populations. And finally, one thing that might be really important, because these are all insectivorous bats, we would want to do invertebrate surveys to see if that can kind of tease apart the reasons why we have differing occupancy between these survey points. And that could maybe be a potential explanatory variable for occupancy of these different species. And so with that, I will take any questions.
Questions, welcome.
I have a question.
Yes, please.
My question is, that map that you showed of the white nose syndrome spreading across the United States. It really didn't, the white nose didn't really spread further than what, South Texas? Is that like, is it more a question of when it goes south, or is there a reason that this like deep south is less affected? Do you have an answer to that?
Did you hear well the question, or you want it to be repeated?
I heard the question. Yeah. Is there a reason that there isn't as much spread in the southern states and into Mexico? Was that your question?
Yeah.
So actually, a lot of the reason for that is just because those areas tend to be understudied. And so we don't have as much data for a lot of those areas. And it's generally considered that the spread is inevitable because, you know, a lot of these species are highly migratory. So as they travel, they're going to keep spreading this disease. And so it's really just another instance of where more research is needed.
Thank you.
Hi, Corrine. Are there any bat facts that you wish everybody knew that might maybe motivate them to, you know, live in service of the bats?
Oh my gosh, that is such a good question. There are so many bat facts that I wish people knew. The things that people might actually care about probably goes back to those ecosystem services. I briefly touched on that in the introduction when I was speaking on how they, you know, contribute billions of dollars a year in the United States. And that's just for pest control alone. So just their agricultural value in providing insect control services on croplands essentially is worth billions of dollars. And I think that would be a really good way to get the general public engaged in conservation work is, unfortunately, to us, you know, because we think that all nature and all wildlife are inherently valuable. For some people who may not be as interested, seeing the economic value and the services that these species can provide is another kind of incentive to get people to care about conservation.
Thank you very much. Welcome. And we can have our next.
Thank you.
Yeah, so hi again, my name is Melissa and I'll be presenting my master's thesis research under my advisor, Madeline Vanderkirk, and my other committee members, Kathy Broadhead with BLM and Jonathan Coop, also with Western Colorado. Sweet. So I just want to start by acknowledging the many people that made this project possible, help from other people in federal and state agencies, field technicians, contractors, other Western students helping with contributing their own research in this area, my lab mates, and obviously my friends and family. But without help from all these people, this project wouldn't have been possible and I wouldn't be able to present this research here today. So to begin with the candidate links, there are habitat and prey specialists adapted to high elevation forests and adapted to areas with deep snowpack and harsh winters. They are listed as a federally threatened species in the US and as state endangered in Colorado. However, they were extirpated from the state by the mid-1970s due to unregulated trapping and habitat loss. As a result, Colorado Parks and Wildlife initiated a reintroduction program, which saw them return to the state in the early 2000s. And ever since, post-release monitoring has continued to protect this population, ensure it's a self-sustaining, viable population. And primarily, a lot of work has been done to identify and protect critical lynx habitat in the state. So snowshoe hares are the primary prey of lynx. The overlap, or the ranges of the two species virtually overlap, and so conservation of these lynx and hares go hand in hand. Throughout their range, their primary habitat is mature multi-storied spruce fir forests. And these, in Colorado specifically, this is composed of Engelmann spruce trees and subalpine fir trees. These stands contain dense vegetation and a high horizontal cover, which provides numerous survival benefits to hares, primarily browse availability, thermal protection, and lower predation risks. So hare densities can be used to evaluate habitat suitability for lynx, and they can be estimated from fecal pellet counts. And so this method, as opposed to direct monitoring methods for lynx, tends to be a lot less time consuming and a lot less costly. Previous research has suggested a minimum hair density of 0.5 hairs per hectare is required on the landscape to support a resident population. However, in Colorado, this may be as low as 0.2 hairs per hectare. Spruce bark beetle impacts have been widespread throughout the Southern Rocky Mountains, however, and so this could be having potential impacts to hare and lynx habitat. Since 2000, over 1.9 million acres of spruce fir forest have been affected in Colorado alone by these beetles. And so with these potential impacts, there's a need to understand, in Colorado, are these severe spruce beetle outbreaks impacting lynx and hares and affecting this reintroduced population? So the Powderhorn Wilderness Area is located here in southwestern Colorado. These forests is co-managed by the Bureau of Land Management, managing the northern portion, and the Forest Service managing the southern portion. This area has seen extensive spruce mortality and loss of mature trees from its forests. If you go out there, you can just see entire hillsides of just dead spruce trees, which it's pretty sad to see. But so this extensive standing dead and down logs contributing high fuel load alongside warmer temperatures and drought, further drying this fuel promotes extreme wildfire risks in this area. And wilderness areas are unique as compared to other types of public lands in that there are no roads and few trails. And the Powderhorn specifically is very remote with really rugged terrain. And so if a wildfire were to ignite, suppression would be extremely difficult. And this threatens not only nearby communities, recreation, but also any remaining critical wildlife habitat. And so as a result, managers have proposed several potential treatment areas to reduce this fuel load and promote regeneration of a healthy habitat for lynx. This area is located only 5 to 10 miles from the core lynx reintroduction area in the San Juan Mountains. And lynx have been documented in the surrounding areas, but none have yet been seen within the Powderhorn. And so this leaves a need to understand if the spruce fir forests in the Powderhorn can actually support a resident lynx population, where this habitat might exist, and if removal of the standing dead beetle-killed trees would impact hares and lynx in this area. So our overarching goal was to evaluate the North Powderhorn Wilderness, an area just north of its boundary, making up the planning area for kin and lynx habitat, and use this information to inform management practices in this area. First, we wanted to estimate hare densities using from fecal pellet counts to determine if this area actually contains enough hares that can support lynx. We then wanted to identify the important parameters of hare habitat from the forest structure and topographic covariates that we collected. And finally, we wanted to evaluate these potential impacts of spruce bark beetles on lynx and hare habitat in this area. So in the late summer and early fall, in 2021, 2022, we conducted 90 pellet count surveys and collected vegetation and topographic data from these survey sites. We conducted surveys both inside of these 3 potential treatment areas and outside of them. And then from these 90 sites, we restricted our analysis only to those that were within the spruce fir elevation zone to remove potential bias and pellet counts from another leproid species that may coexist at lower elevations. And so this left us with 79 sites for our analysis. So for our data collection at each survey site, we established a permanent circular survey plot centered on a rebar stake. And then we had 2 transects running north, south, and east, west, crossing out the plot center. We then established 5 pellet subplots along the north-south transect. Within each of these, we counted and removed every single hare pellet within the plot boundary, and we classified them as new or old based off the color, shape, and texture. And so because we count and clear these pellets, these surveys are typically conducted over a 2-year period, with that first year survey being under uncleared conditions. So that way, when the pellets are recounted, this has an estimated hare density of over a known 1-year period. However, these studies are currently ongoing, and so this study focuses on the uncleared pellet counts. So a lot of pictures for our data collection. We also, in the pellet plots, estimated the percent ground cover by grasses, forbs, and shrubs. We calculated the density of down logs, as well as the understory tree stems. We estimated the percent canopy cover using a moose horn and percent dense horizontal cover by vegetation, logs, and rocks using a cover board. We then calculated the tree density as well as the tree basal area of all mature trees over 3 inches diameter. And we separated these into live and dead trees for each of the 4 dominant tree species. And we included metrics of tree density in addition to basal area, as basal area takes into account the diameter of the tree and so the maturity of that tree. Then for our topographic covariates, we extracted values at the plot centers for elevation, slope, or how steep it is, and the terrain ruggedness index, so how rough or flat that plot is. And we took this from a digital elevation model from the USGS. So to calculate hair density, we used 2 different equations developed in Colorado, one for uncleared conditions and one for cleared conditions. And we use this calculated hair density in the powder horn from counts of new pellets. And so we estimate that the true hair density in the powder horn will lie somewhere between this range. So then program R, wonderful software. We determined that use a model selection approach to determine the most important predictors of hair habitat in the powder horn. And we ranked and compared models based off the AICC value. And then in addition, we calculated the relative variable importance. And so this is used to determine which independent covariate is considered important based off of a level of 0.5 or greater. And then to look at impacts of bark beetles on these forests, we had 2 primary questions. To answer the first, we compared spruce tree density and spruce basal area between live and dead trees. And then we repeated our model selection approach again, but only within 25 stands that were entirely composed of spruce trees to really get at, do components of live and dead spruce trees affect hair habitat selection? And so now for some results. We first used these 2 density equations to crosswalk hair pellet counts to densities. Our lower estimate was 0.1 hairs per hectare, and our upper estimate was 0.18 hairs per hectare. And so again, we assume the true density in the powder horn during this survey period to be within this range. Our model selection indicated that the best predictors of hair habitat was high horizontal cover, live spruce fir basal area, and elevation. And so this model indicates that high quality hair habitat has dense vegetative component, more large diameter trees, specifically Engelmann spruce and subalpine fir, and at lower elevations, but within this spruce fir elevation range. Our relative variable importance showed that horizontal cover was the only covariate considered important at that 0.5 level. It had a positive relationship with the new pellet counts. And so you can see here in the pictures that hares are preferring habitats with much more vegetation, much denser, which provides those benefits to hares that I mentioned before. Looking at mature spruce trees in the powder horn, we didn't find a significant difference here in the spruce density between live and dead trees shown on the left. However, we did find a significant difference in the basal area with significantly greater basal area of dead spruce trees than live. And so this further indicates that these mature trees are being lost from spruce fir forests in the powder horn. Looking at hair habitat selection in pure spruce stands, we found that live Engelmann spruce basal area was the best predictor of hair pellets, and again, had a positive relationship with these pellet counts. And this further emphasizes the importance of this specific tree and the maturity of these trees for hares. And you can see here, the smaller trees have much less basal area, the regenerating versus mature trees. And I don't actually have a photo myself of a mature Engelmann spruce tree because we found none in our survey sites. So it's a little depressing, but. And then just to place our results into management implications. And comparing to similar studies in Colorado, our hair pellet counts were similar to those reported in areas without links, but were lower than those counts reported in areas that are currently occupied by links. Similarly, our hare densities were comparable to the lowest ranges estimated in other spruce fir habitats in Colorado, currently occupied by lynx. And then further comparing our density estimate to these proposed thresholds, even if this threshold is as low as 0.2 hares per hectare in Colorado, our estimated density still falls well below this range. And so this overall suggests a lack of suitable habitat in the Powderhorn for lynx and likely cannot support our resident population in the planning area. So our model selection again identified these three components to be representative of primary hare habitat in the Powderhorn, and research across the species range agrees with our findings, especially that horizontal cover is the most important parameter, as it provides numerous benefits to hares. And interestingly, horizontal cover has been found to even offset impacts of beetle kill to hare and lynx habitat use by maintaining these small patches of habitat refugia that can be used by these species. While these bark beetles aren't necessarily directly impacting hares and lynx, however, they've listed a high mortality of Engelmann spruce trees, which we identified to be very, very important for hares and thus for lynx as well. And so this indicates that these severe spruce bark beetle outbreaks in the Powderhorn could be having potential indirect impacts to lynx and hares via the loss of this mature forest, these mature spruce trees. And this is especially true if these trees cannot regenerate, as mature trees are required to supply that cone source, that seed source. So especially if that understory cannot regrow, then these forests will no longer have any lynx or hare habitat remaining. And so again, this extensive standing beetle killed trees and down logs alongside prolonged regional drought and high temperatures has led to high risks of severe wildfires in this area, which we, like I said before, are very, very difficult to suppress. And so this threatens not only nearby communities and recreation, but any remaining lynx habitat. So this further just demonstrates the importance that active stand management could be extremely useful to reduce this dangerously high fuel load. And appropriate treatments can also create fine-scale habitat heterogeneity while promoting the regeneration of a healthy spruce fir forest with high horizontal cover. Treatments that can retain any residual live vegetation, especially any remaining live spruce trees and dense cover, understory growth, can create a mosaic of habitat types and promote understory regrowth. And this can be used to meet both management goals as well as conservation of these two species. However, it is important to note that while these treatments have been shown to benefit hares and lynx in the long term and can create that healthy spruce fir habitat, any treatment will reduce habitat in the short term before it can create it. Not sure if there's time for questions.
One question. Hey, good job, Melissa. Thank you. I was just curious, you didn't mention anything about the 10-year sort of cycle of hares, which occurs across a lot of their range. Maybe hasn't been demonstrated for sure down here, but could these really low population densities of hares that you found be associated with just the low period of the cycle? And they, of course, can go up 10 times higher.
Yeah, so the question was, is these low hare density estimates just a product of being at the low phase of the hare cycle? If I'm getting that correct. Yeah, so in Colorado, found research that suggests that these populations really aren't that cyclic in the southernmost range periphery, which is Colorado. And that they're comparable to the lowest population densities and abundance at that low phase in the northern regions. So yeah, I don't think it's going to change much from year to year, even like a 10-year period, just because they really aren't that cyclic. They're much more stable. Also, the greater use of alternate prey species, such as red squirrels, primarily in Colorado, can really offset any impacts of areas with low hare abundance. And that's especially true for Colorado. I think one study found that like red squirrels can make up as high as like 8% of prey biomass in lynx winter diet in Colorado. However, red squirrels are more negatively impacted by spruce bark beetles because they no longer have that cone source for food. And so, I mean, I didn't necessarily go into that for this project, but that's just another threat to lynx because not only are these hair densities much lower than we would expect them to be to support population, but they're lacking that alternate prey source as well.
Thank you very much. Julie, welcome.
Whew, it's quite a thing to follow, and unfortunately, I don't want to say unfortunately, I'm presenting a less gregarious pollinator, nowhere near as cute as the bat. And I also recognize that it's the lunchtime hour, so I've cut out the math and I've added a lot of flowers and butterflies to my presentation and a lot fewer words. So I am presenting the resource selection by an endangered butterfly. And a bit of background is that climate change is driving shifts in alpine floral phenology, distribution, and composition, and that's resulting in direct and indirect impacts on pollinators and population dynamics of those pollinators. So basically what that means is flowers are blooming early, flowers are blooming late, flowers are shifting in elevation, and the mix of what flowers are where is changing. An example of one of these effects is the mismatch of historically synchronized events, such as if a pollinator were to emerge prior to its floral resource, the floral resource that it has used historically. And this can lead up to 50% of pollinator species facing disruption to their food supply. Principally, that will happen at the beginning or at the end of their activity period. Oh, and sadly, and I had my sad larvae here, that may result in the loss of some interactions, potentially novel interactions, like a butterfly visiting a flower it's never visited before, but also it can result in a decline in populations and potentially even extinctions of pollinators. And also, they're seeing that there is an elevational shift of flowers. So Zorio et al. Saw over a 65-year period that in the Rocky Mountains, some flowers were shifting upwards of 41 meters. And that's quite a range shift. And that's, again, leading to a new composition of flowers at different elevations. So here's my adorable research organism, the Uncompahgre fritillary butterfly, which I will hereafter call the UFB. It is a federally endangered species. It's endemic to the San Juan Mountains of Colorado, so not far from here. And it's only known to exist in 11 alpine colonies, somewhere between 3,800 and 4,100 meters. So they're living in alpine tundra. The UFB is a specialist on its larval host, which is the dwarf shrub snow willow, Salix nivalis. And you'll see a picture, that's the UFB on its host plant there. And the UFB is very small, and so is its host plant. But it is considered to be a generalist on nectar as an adult. Interestingly, though, Salix nivalis kind of covers the floor all over the region, but the UFB isn't found within the entire range of its host plant. That's an indication that it has other habitat requirements, and we're still trying to figure out what those habitat requirements are. Also, since 2003, there have been annual population surveys of the UFB. So last year, I was also part of the annual population survey to determine the persistence of the UFB, as well as the number of UFB. And a little inside note is that we determined there were about 7,500 UFB this past year, which is the best number since 2017. So hopefully their population may be going up, or it may be cyclic. The objectives of my study were firstly to identify the nectar resources used by the UFB and to rank them from most to least selected, to determine the nectar quality available within the UFB's habitat before their flight period, during their flight period, and after their flight period, and also to determine if the nectar availability differs before, during, and after their flight period. In order to do this, The best part was I got to observe the butterflies. And I spent about 12 hours in 5-minute bursts following the butterflies around the alpine tundra and recording basically the flowers that I saw them nectaring on. And there was a strategy to that. And this was only under suitable conditions. They don't like to fly if there's any clouds or any rain, so it had to be a beautiful sunshiny day with low winds and not too cold and not too hot. The second part of my methodology was to assess the floral resources. So basically I established plots across the entire habitat, and then I visited those plots every day and I counted the number of flowers in every single plot. And there were many flowers because most of these flowers are very tiny. And the plots were determined by the historically highest and lowest number of UFB in that area. And lastly, I extracted nectar from the flowers within the habitat. And I selected 2 flowers of every species every time I visited the colonies. So that was about 6 to 8 species, or 6 to 8 flowers of each species every day. I sampled those prior to the UFB activity. That's called the standing crop nectar availability. So that's kind of like the nectar that's available to the UFB, ideally. And in order to extract them, I either use these tiny microcapillary tubes that just suck the nectar out. I used a centrifuge, or more often than not, I had to bring my nectar samples back here to Western and analyze them in the lab. And basically what I did is I used a spectrophotometer to look at the absorbance, and that told me the nectar sugar content in that nectar sample. And I did a whole lot of math. I also used R, and I used compositional analysis, which basically looks at the species that are used to the availability of that species, and that ranks those species from the most to least selected. And I also used a few other tests to determine if there was variation in the nectar, interspecific, or community level. So like, between that species within itself, between the different species, and then at an entire community level. So what did I find? Well, I did 142 observation periods, and in 112 of them, I saw the UFB nectaring on a flower. Within those 112 nectaring events, they visited 179 flowers of 15 different species. And there were 41 species that I sampled within the habitat. So they used 36.59% of the flowers within the habitat. But what I found most interesting is those 15 species actually represented 76.7 to 99.98% of the nectar within the habitat. So basically they were like riding the green wave. They were just consuming the most amount of nectar that was available within the habitat. The compositional analysis indicates that the resources used by the UFB were used selectively rather than in proportion to availability. So some flowers may have been very rare, but they kind of were like, yeah, that's the good stuff. And those UFB went to those flowers. They were really choosing the flowers that they wanted, probably the ones with the most nectar. I sampled 547 different flowers. And this is just a chart of the number, just showing the 15 species that were selected by the UFB, the number of flowers of each species that I sampled, and then SU is the sugar, the average sugar per unit. And I also included there the maximum and a minimum amount of sugar in those flowers throughout that period of sampling, which was actually June 5th through August 14th, 2022. And I saw a range of 0.0013 milligrams of sugar in a single alpine clover flower to as much as 4.57 milligrams of sugar in Old Man of the Mountain, which is the picture on this slide. And lastly, here's just a fancy graph showing that there was nectar variation between the different time periods, pre-emergence, during the flight period, and post-flight period. So what did I find? Basically, I found that the UFB is a generalist, but it is selective in the flowers that it is choosing. And there were observations from previous years that have indicated that the UFB did select flowers that I didn't observe them selecting this year. And that shows me that there's probably some flexibility in their foraging behavior, especially because the two of those flowers occurred prior to the UFB's emergence this year. So that means that it's possible that like flower phenology shifted, and that year those flowers were more available then, and this year they weren't as available. And I also saw that there was variation in the food resources. So some flowers emerged and then they disappeared, new flowers emerged, and that kind of indicated what flowers that the UFB was selecting at that time based on what was available to them. Again, the UFB was riding the green wave. It was consuming the most amount of nectar available within the habitat. Prior to their emergence, the nectar from the flowers that they selected was only 17.63%. And then once they disappeared, it dropped back again to 40.16% of the nectar sugar in the habitat. So what does this mean for management? Firstly, a knowledge of the host plant and the adult resources used by this endangered butterfly help us understand where the UFB is going to be in the first place, because it's most likely that the UFB will shift its location with its resources. Most importantly, though, I think, is that the knowledge of the adult resources and the host plant is going to help us prioritize climate refugia and dispersal corridors. One of the most important aspects of this, for such a small population, is divided into subpopulations. However, in order for the UFB to travel, it has to kind of find suitable habitat patches in between the subpopulations. And that is critical for gene dispersal and for the persistence of the species. And of course, if we ever have to restore the species or reintroduce the species because it is endangered, we obviously want to know what suitable habitat looks like. So my conclusion is that the UFB's plasticity in foraging behavior and historic observations of its use of different species indicate that it may have the potential to buffer indirect and direct impacts of climate change. I'd just like to acknowledge, of course, my committee members. That's Dr. Carol Boggs and Dr. Jane Ogilvie, Kathy Broadhead, and Dr. Kevin Alexander. And thank you to Uncompahgre, Gunnison, Rio Grande, and San Juan National Forest, the U.S. Fish and Wildlife, and the BLM Gunnison Field Office, as well as Western for supporting this research. And I have lots of literature. Thank you.
Well done. One question from the audience? Well, if there are no questions, yeah.
Did you experience or encounter any lynxes in your science? Oh, this is for the group.
I did not. I wish.
Can you repeat the question, please?
I have a butterfly question. I'm sorry. Do the butterflies ever like outcompete each other for one resource?
You know, that was a theory I had. Within the species, I don't think so. But I was kind of curious because these butterflies start flying after a lot of other insects are already flying, that they might be getting the short end of the stick. However, flowers do tend to produce nectar throughout the day. So that would provide plenty of nectar for them.
And
I forgot to answer the other question, sorry.
No problem. But we are going to move to the general questions for the whole panel. And then there will be also chance to continue with the questions for Julie. Yeah. Well, first question, yeah.
My question is for Julie. How often, if at all, do flowers regenerate their nectar?
And if you
She's
can
asking
repeat
how,
the question, please.
Yeah, how often, if at all, do flowers regenerate their nectar? They do it, I believe, all the time. So a lot of other studies use exclusion bags, like bags they put over the flower, so nothing, nothing nectars on that flower, and then they can sample the flower. So definitely overnight, they will re-up their nectar. Yeah.
I have a question about lynxes. Have you experienced any lynxes or encounter them, and why are their feet so big?
Well, we didn't, I mean, I haven't, oh, sorry. So the question was, yeah, have I seen any lynx out there? And why are their feet so big? For the first part of the question, yeah, I haven't seen any lynx out there. There have been camera trap surveys in the area since 2014, and still none have been documented, but they're within the surrounding areas. So it's possible that they're within other areas of the Powderhorn. It's a very, very big area, but we were sampling particularly in beetle impacted, severely impacted stands. So it makes sense why we didn't see any. But as to why their feet are so big, they act as snowshoes. So that way they can access high elevation habitats with really deep snowpack. And so that lets them outcompete other predators such as coyotes and mountain lions for snowshoe hares at those higher elevations. But yeah, they're like the Bigfoot of this area.
Yes.
I have a question for Julie. Of the plants that you surveyed, did you find that your species was using any plant specifically to grow, like lay their eggs on, or like was there a preferred plant for the larva?
Yes, the question is, did the butterfly have any specific plant for laying its eggs on or for the larvae? And yes, that was already known that its host plant, Salix nivalis or snow willow, is the ovipositional substrate where they lay their eggs, and it's what the larvae eat. I will tell you though, I followed all these butterflies for many hours and I tried so hard and never saw a single butterfly lay an egg. So I'm curious who knows that and how they figured it out, because there hasn't been. There are only so many papers on this one specific species.
This is kind of for the panel, like all of your work is really amazing. Just kind of, do any of you have contingencies in place, like someone to take over your research? Are you continuing on? Just kind of what you expect from now on. If you have
Well, for the Uncompahgre fritillary butterfly, there are still annual population surveys. So I think that is to be continued, but I don't believe there's another grad student on the project for next year. And we'll see what the managers decide to do in terms of continuing the endangered species monitoring.
Yeah, for the Powderhorn, these surveys are being repeated this year and then will be continued in following years to assess these snowshoe hare populations and reassess for Canada lynx habitat. And this is funded by the BLM, and it's a BLM project trying to help inform these treatment areas and see, as part of the Endangered Species Act, BAs, and just making sure that if they do treatments, it's not in areas that are habitat. So yeah, this is just ongoing and will continue to happen.
Corinne, the question was if you have any continuity plans. Did you want to touch on that?
Yeah, thanks, Brandon. So most of my survey sites were on Forest Service land. So I'm trying real hard to get the Forest Service biologists in the Gunnison National Forest to pick up this project. It does require a lot of specialized equipment and expertise, but a lot of the other local units, like for the BLM and the Park Service, already have that. And so my hope is to get either another student researcher to continue this project, or to get a Forest Service biologist or like a seasonal technician to continue the monitoring each year.
Cool. And I would say for the owls, the BLM is planning to use the passive acoustic monitoring, but also we're helping to contribute our occupancy data to the Forest Service, who is making this habitat suitability map across the entire state of Colorado, so that we can try to go through and see where else there might be owls across the whole entire state.
There is a question on Zoom. Andrea Williams is asking Julie, which flower species had the highest nectar sugar content?
Hi, Andrea. Yeah, definitely Old Man of the Mountain was the top. And then Elephant Head, and you'd probably be interested in this, I'm talking to Andrea, was probably had the next most, because I was looking at the entire plant, which was about 50 flowers. And interestingly, it was one of the most preferred species selected by the UFB was the elephant's head, which has a really weird, I don't know, a weird physical appearance. So.
Got a question for Julie as well. What were the flowers when you were like gathering the nectar amount? Were you also testing the pollen content of the flowers?
No, I wasn't.
And if you can repeat the question, because our audience, we have some noise from the air conditioning, and for the distant audience, please.
The question was when I was sampling nectar, if I was also sampling pollen, and I didn't get into pollen because UFB aren't known to consume pollen. So I actually, when I had a centrifuge, I would kind of cut the pollen out of the flowers.
Last.
My question is for Corrine. I'm wondering what causes the white nose disease?
Okay, so the question was, what causes the white nose disease? It is this fungus called Pseudogymnastus destructans, and we don't really know where it came from. It just kind of appeared in the United States in 2006 and just spread super rapidly. Further research and testing has found that it's also present in Asia and Europe, though it doesn't have the same like epidemic devastating qualities that it does here for some reason. And scientists are currently researching that. But basically what it does is it's this fungus that infects the bat's respiratory system, and you can literally see it. It looks like a white, just like fuzz on their face, and it spreads to the rest of their body. It starts on their nose, so it's called white nose syndrome. And so it'll infect them and spread during hibernation, which is obviously when they're supposed to be in torpor and they're supposed to have a really low metabolic rate, and it wakes them up during hibernation. And so it really screws with their metabolism, and they essentially die from either starvation or dehydration during the hibernating period. It's pretty crazy.
Well done for this panel.